Good morning. We start off with the next topic on biochemistry and chemistry titled RNA synthesis and processing. Here we will learn more about the different types of RNA and how RNA is transcribed from the DNA sequence. We will start with section 10.1 covering the types of RNA. Right, so DNA versus RNA, we have deoxyribose in DNA, ribose in RNA, thymine in DNA, uracil in RNA. Right, recall that RNA has two chemical differences from the DNA molecule. RNA uses the ribose sugar that contains 2 OH and it uses the uracil base in place of thymine. We'd like to thank the Western Oregon University for this invaluable notes that students wish to follow. So with all due credits to that university and their notes, I'm reading it out for all those students who can't afford to study by spending that kind of heavy fees for their studies. So please listen as clearly as possible and be attentive, take as many screenshots and make your notes, and wish you all the best. We go ahead with the subject proper from now on, right? Best wishes. So this is DNA versus RNA. These are screenshots that I suggest you please take while I teach, right? And read out the matter for you. These minor chemical differences result in major structural differences between the molecules. While DNA is held in the rigid structure of the alpha helix, RNA is usually single-stranded and therefore has much more variation in the shapes that it can adopt. RNA can largely be divided into two types: one that carries the code for making proteins or coding RNA, which is also called a messenger RNA, mRNA, and non-coding RNAs, ncRNA. The ncRNA can be divided into several different types depending either on the length of the RNA or on the function. Size classification begins with the short ncRNAs, 20 to 30 nt, which include microRNAs, MIRs, and small interfering siRNAs. The small ncRNAs up to 200 nt, which include different transfer RNA, tRNA, small molecular RNA, snRNA, and small nuclear RNA, snRNA, and long cRNAs. Greater than 200 nt, which include ribosomal RNA, rRNA, enhancer RNA, eRNA, and long intergenic ncRNAs, link RNAs, link cRNAs, among others. Right, and continuing onward, into the next page, we see what's translation about. We see this figure there. The amino acids, ribosomes, large subunits. We have a translation of a Gron's peptide chain with the tRNA and mRNA, rendering the procedure viable, right? So, going into the subject matter proper there, cells access the information stored in DNA by creating RNA through the process of transcription, which then directs the synthesis of proteins through the process of translation. The three main types of RNA directly involved in protein synthesis are messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. The mRNA carries the message from the DNA, which controls all of the cellular activities in a cell. If a cell requires a certain protein be synthesized, the gene for this product is turned on and the mRNA is synthesized through the process of transcription. The mRNA then interacts with ribosomes and other cellular machinery to direct the synthesis of the protein it encodes during the process of translation. mRNA is relatively unstable and short-lived in the cell, especially in prokaryotic cells, ensuring the proteins are only made when needed. And this is microRNA, miRNA, right? And the post-transcriptional expression levels of many genes can be controlled by RNA interference, in which miRNA-specific short RNA molecules pair with mRNA's regions and target them for degradation. This process is aided by protein chaperones called argonauts. This antisense-based process involves steps that first process the miRNA so that it can base pair with a region of its target mRNAs. Once the base pairing occurs, either proteins direct the mRNAs to be destroyed by nucleases. Farmelo awarded the 2006 Nobel Prize in Physiology or Medicine for this discovery, right? So I'd like to read that again for you. This antisense-based process involves steps that first process the mRNA so that it can base pair with a region of its target mRNAs. Once the base pairing occurs, other proteins direct the mRNA to be destroyed by nucleases. Fire and Mello were awarded 2006 Nobel Prize in Physiology and Medicine for this discovery. And the next diagram would be that of types of RNA molecules, where you see RNA, tRNA in the RNA by mass and RNA by number of molecules. It's a difference for RNA and tRNA. Please take screenshots, and you need to make notes there again. At steady state, the vast majority of human cellular RNAs consist of rRNA. At 90% of total RNA for most cells, figure 10.5. Although there is less tRNA by mass, their small size results in their molecular level being higher than RNA. Figure 10.5, other abundant RNAs such as mRNAs, snRNA, and snORNAs are present in aggregate at levels that are about 1 to 2 orders of magnitude lower than rRNA and tRNA, figure 10.5. Certain small RNAs such as miRNA and piRNAs can be present at very high levels. However, this appears to be cell type dependent. LNC RNAs are present at levels that are 2 orders of magnitude less than total mRNAs. Although the estimated number of different types of human LNC RNAs may have a very restricted expression pattern and thus accumulate to higher levels within specific cell types. For example, sequencing of mammalian transcriptomes has revealed more than 100,000 different INC RNAs or LNC RNA molecules can be produced. Compared with the approximate 20,000 protein coding genes, the diversity and functions of the transcriptome within biological processes are currently a high active area of research. Here in transcription and RNA processing, we have RNA polymerase enzymes. RNA polymerase enzymes are required to carry out the process of transcription and are found in all cells ranging from bacteria to humans. All RNAPs, RNA polymerase enzymes, are multi-subunit assemblies, with bacteria having 5 core subunits that have homologs and archaeal and eukaryotic RNAPs. In this section, we will learn more details about this enzyme class. Right, so this is the transcription initiation. Please take screenshots and I'll elaborate the notes here. Transcription takes place in several stages. To start with, the RNA polymerase holoenzyme locates and binds to promoter DNA. At this stage, the RNAP holoenzyme is at the close conformation RP. Let's see. Initial specific binding to the promoter by sigma factors of the holoenzyme sets in motion conformational changes in which the RNAP molecular machine bends and wraps the DNA with mobile regions of RNAP playing key roles. Next, RNAP separates the 2 strands of DNA and exposes a portion of the template strand. At this point, the DNA and the holoenzyme are said to be an open promoter complex, RP, and the section of promoter DNA that is within it is known as a transcription bubble. This is a diagram of the RNAP, and we continue with the subject matter proper. The RNAP catalytic core within bacteria contains 5 major subunits: alpha 1, beta, beta 1, that's omega. I presume that's omega, yeah. To position this catalytic core onto the correct promoter requires the association of a 6th subunit called the sigma factor, sigma. Within bacteria, there are multiple different sigma factors that can associate with the catalytic core of RNAP that help to direct the catalytic core to the correct DNA locations where RNAP can then initiate transcription. For example, within E. coli, sigma is the housekeeping sigma factor that is responsible for transcribing most genes in growing cells. It keeps essential genes and pathways operating. Other sigma factors are activated during certain environmental situations, such as sigma, which is activated during starvation, or when cells reach the stationary phase when the sigma subunit associates with the RNAP catalytic core. The RNAP has then formed the holoenzyme. When bound to DNA, the holoenzyme conformation RNAP can initiate transcription. Once the transcription bubble has formed and transcription initiates, the sigma subunit dissociate from the complex and RNAP catalytic subunit continues elongation on its own. That is the eukaryotic RNA polymerase enzyme, right? And in eukaryotic cells, 3 RNAPs share the task of transcription. The first step in the gene expression, RNA polymerase 1 or pol 1 is responsible for the synthesis of the majority of RNA transcripts, where RNA polymerase 3, pol 3, produces short structured RNA such as tRNAs and 5S RNA. RNA polymerase 2, that's pol 2, produces all mRNAs and most regulatory and untranslated RNAs. The 3 eukaryotic RNA polymerases contain homologs to the 5 core subunits found in prokaryotic RNAPs. In addition, the eukaryotic pol 1, pol 2, and pol 3 have 5 additional subunits forming a catalytic core that contains 10 subunits. The core has a characteristic crab claw shape, which encloses a central cleft that harbors the DNA and has 2 channels, one for the substrate NTP and another for the RNA product. 2 pinchers called the clamp and jaws stabilize the DNA at the downstream end and allow opening and closing of the subcleft for transcription to occur. The enzyme has to maintain a transcription bubble with the separated DNA strands, facilitate the addition of nucleotide translocate along the template, stabilize the DNA, RNA hybrid, and finally allow the DNA strands to re-anneal. This is achieved by a number of conserved elements at the active site, which include the fork loop or loops, rudder, wall trigger loop, and bridge helix. The next diagram shows pol 1, pol 2, pol 3, and pol 3 and the pol 2 picture, right? So we read on the subject matter there. Here you can see a comparison of all 3 RNA polymerases from eukaryotes. They are all homologs and share a similar core complex shown in the gray and tan regions. The stop region is also a related structure in all the polymerases. The pol 1 and 3 enzymes have more subunits than pol 2. They have more limited arrays of transcripts than they create and tend to permanently incorporate transcription factors into their core structures. Pol 2, however, has a much more diverse array of targets and therefore has a wide array of transcription factors that it needs to bind with. Therefore, they are not permanently incorporated given a smaller polymerase structure. In the next section, we'll focus on the activity of polymerase 2. Right? This is about transcription and RNA processing, where transcription factors and the pre-initiation complex will be dealt with. RNA transcription consists of 3 phases: initiation, elongation, and termination. In this section, we will focus on the initiation phase of transcription, where RNA polymerase 2 is recruited and activated by a host of transcription factors. Now, the general transcription factors, the TFID, TFRIH, TFIF, the pol 2, TFRE, the TFRA, and TFI2B, all right? Co-promoter DNA, and the sequential assembly pathway. Now, transcription factors that are critical for the recruitment of RNA polymerase, right? 2 are typically referred to as the TF2 class of transcription factors. They are further identified by a letter A, B, and C, and so on. Class 2 gene transcription is regulated at various levels, while assembling on chromatin before and during transcription initiation, throughout elongation and mRNA processing, and termination. A host of activators and pressors has been reported to regulate the transcription, including a central multi-subunit complex called the mediator that helps the recruitment of general transcription factors, GDFs, and the activation of RNA pol 2. Here we will focus on the picture formation of the GDFs, that are general transcription factors, that make up the core pre-initiation complex, PIC, during the transcriptional activation. The pre-initiation complex PIC is shown here. Take a screenshot and go ahead with the notes where you like. Get them across to you. Transcription of RNA pol II dependent genes is triggered by the regulated assembly of the pre-initiation complex. Peak formation commences with the binding of TF2D to the core promoter. TF2D is a large megadalton-sized polyprotein complex with around 20 subunits, made up of 14 different polypeptides. The Tara-box binding protein, TBP, and the TBP-associated factors, TAFs, numbered 1 to 13. TF2D was shown to adopt an asymmetric horseshoe shape with 3 almost equal-sized lobes, A, B, and C, exhibiting considerable degree of conformational flexibility, with at least 2 distinct conformations, open and closed. This is the illustration of the TBP, the Tara-box binding protein, right? The TBP Tara-box binding protein is a component of TF2D, binds with the specific DNA sequence called the Tara-box. This DNA sequence is found about 30 base pairs upstream of the transcription start site in many eukaryotic gene promoters. TBP Tara-box binding protein. If inhibitors bind the TBP, it will be unable to interact with the DNA and start the formation of pre-initiation complex. The Tara-box binding protein. In the absence of inhibitors, TF2D will bind with the DNA and the Tara-box binding protein, which scan the DNA for the Tara-box sequence. The TBP Tara-box binding protein, which is engaged here. In the absence of inhibitors, TF2D will bind with the DNA and the Tara-box binding protein, which scan the DNA for Tara-box sequence. When TBP binds to a Tara-box within the DNA, it distorts the DNA by inserting amino acid side chains between base pairs, partially unwinding the helix and doubly kinking it. Note that TBP is released from TF2D during the process. This is TBP again, Tara-box binding protein, right? Blocking off TF2B binding site. The diagram on the right shows the TF2D complex in scanning mode. That is here. You see this one in scanning mode. Notice that several transcription factors binding sites are blocked during this time so that the pre-initiation complex won't form inappropriately when TBP binds with the Tara-box and dissociates from TF2D. This opens up a binding site for TF2B, which will in turn bind with RNA polymerase II and initiate the formation of the pre-initiation complex. Pre-initiation complex is shown here, right? So take screenshots and take down my notes. This slide shows just this just shows the assembly a little more clearly. Okay, TF2D binds to the DNA when the TBP finds a Tara-box. It releases from TF2D and can recruit TF2B and the RNA polymerase II enzyme. In addition to other TF2 factors, TF2D may dissociate completely or stay loosely associated with the TF2A. The TF2B is critical for the transition of RNA pol II from the closed to the open conformation, and transcription can begin. In most eukaryotes, after synthesizing about 20 to 100 bases, RNA pol II can pause, promoter proximal pause, and then disconnect from promoter elements and other components of the transcription machinery, giving rise to a fully functional elongation complex and a process called promoter escape. The promoter-bound components of the PIC, in contrast, remain in place, and thus only TF2B, TF2F, and RNA pol II need to be recruited for initiation, significantly increasing the transcription rate in subsequent rounds of transcription. Enhancer elements. Right, these are the enhancer elements: transcription complex assembly, initiation, termination, transfer, and the rest of the details as mentioned here. Now, the enhancer elements are also important for the initiation of transcription. Promoters are defined as DNA elements that recruit transcription complexes for the synthesis of coding and non-coding RNAs. Enhancers are defined as DNA elements that positively regulate transcription at promoters over long distances in a position in a orientation-independent manner. Most of the time, enhancers will bring the RNA polymerase II complex to the target gene area to mediate transcription, and the mediator complex shown in gray helps the looping process occur. However, sometimes enhancers may also lead to the short transcription of non-coding RNAs from the enhancer region, as well as the function of these very short non-coding RNAs is not known. The next would be RNA processing transcription and RNA processing transcription elongation and termination. In this section, we will focus on the elongation and termination phases of transcription. For the elongation material, we will only focus on prokaryotic mechanisms as they are better understood. For termination processes, we will first focus on prokaryotic systems and then take a brief look at the complexity of eukaryotic termination processes. This is a picture of the prokaryotic transcription elongation. Once transcription has been initiated, the elongation phase is where the RNA polymerase enzyme will use the template strand of the DNA to create the nascent RNA. This diagram shows the catalytically active RNA polymerase. You can see that within the enzyme catalytic center, a small transcription bubble forms where the DNA helixes are mounted. The template DNA strand is shown in blue. The magnesium 2 cofactor is shown in red, positioned at the catalytic active site. Right? Red there. Right, and we move on to the next diagram. That's prokaryotic transcription elongation. Please remember to take screenshots. This diagram shows that catalytic activity of the polymerase in a little more detail. The DNA template is shown in gray and the nascent RNA strand shown in red. The polymerase has two major conformations during the process of elongation. A closed catalytic conformation that is used when the polymerase is adding nucleotides to the nascent chain. Notice here that the diphosphate is cleaved from the incoming nucleotide. Further hydrolysis of the diphosphate will release energy that helps to drive this reaction forward. Once the incoming nucleotide has been added, the polymerase has to then translocate down the DNA template to open up the position for the next nucleotide to enter. It does this by switching to an open conformation that is more flexible. This allows the polymerase to essentially pull itself along the DNA template one nucleotide at a time. The next diagram is that of the prokaryotic transcription elongation. So you have the closed Mg catalytic, and within the catalytic center, a bridge region shown in yellow and a hinge loop shown in pink are required for catalysts and translocations. In the closed conformation, the polymerase is modestly flexible, which enables the positioning of the Mg2 cofactor to facilitate nucleotide addition. Following nucleotide addition, the bridge assumes an open conformation that is much more flexible. Movement of the hinge loop causes bending of the bridge, and the polymerase can shift down on the DNA template. This is the transcriptional backtracking illustration. Sometimes the elongation phase does not move in a forward position. The elongation complex can sometimes regress in a process called backtracking. This can sometimes be caused by misincorporation event or by a brief pause of the RNA polymerase complex. The backtracking process, however, stalls the elongation process, and it must now be rescued by additional protein helpers. Right? And now we go to the next one, is the grief factors to the rescue. So this is about grief factors are involved in rescuing the stalled transcription elongation complex. During normal elongation, the GRE protein is bound to the active elongation complex, but does not exert any activity on the complex. It essentially stays out of the way, like a tool in a tool belt. However, upon backtracking or nucleotide misincorporation, the GRE factor produces its own trigger loop domain, which supplants the normal catalytic trigger loop of the RNA polymerase enzyme that we saw in more detail in the previous slide. The cofactor will then cleave off any overhanging mRNA from the backtracked complex. The elongation complex is then rescued and the former normal trigger loop domain from the RNA polymerase will again be functional and can resume the elongation process. This is the illustration of the prokaryotic transcription termination. Right, and so the termination of transcription in prokaryotes can occur using an intrinsic model. The diagram on the left shows the polymerase as in yellow, in the open conformation with the DNA in blue and the nascent RNA in red. DNA in blue, right? The RNA in exit, and the red would be nascent RNA in red, and the DNA is in blue, while the polymerase is yellow. All right. This sequence, so intrinsic termination occurs at specific template sequences. An inverted repeat followed by a run of A residues. This sequence causes the formation of a short-term loop structure in the nascent RNA, which is in essence will derail the polymerase from continuing. Prokaryotic transcription termination. Extrinsic factors can also be involved in the termination process. This can be most aptly seen by the functioning of the RHA protein as the nascent mRNA extends outward from the RNA polymerase machinery. The RHA protein is a small horseshoe-shaped protein that can clamp onto the extending nucleic acid. Biochemical and structural data suggest that RHA initially binds to RNA in an open, lock-pusher conformation that closes into a planar ring as RNA transfers to the central cavity. There, the ssRNA contacts an asymmetry secondary binding site, SBS. Upon hydrolysis of ATP, the ssRNA is pulled up upon conformational changes of the conserved Q and R loops of the SBS, leading to RHA translocation and ultimately promoting RNA polymerase RNAP dissociation. Next is the eukaryotic termination. This be a termination pathway, and in eukaryotes, the termination of protein coding gene transcription by RNA polymerase 2 pol 2 usually requires a functional polyadenylation PA signal, typically a variation of the AAU AAA hexamer. Nascent pre-mRNA is cleaved by the YSH1 protein and then polyadenylated by the CPA proteins. Currently, the details of this mechanism have not been fully elucidated and multiple models have been postulated. So, more details on this chapter, the next section, where we would discuss eukaryotic mRNA post-translational processing in more detail. And this is to do with the transcription RNA processing, where we have processing of the RNA. In this section, we will focus on the major post-transcriptional modifications completed in the process of prokaryotic and eukaryotic messenger RNA. Prokaryotic mRNA processing. Watch these and these. And bacterial cells do not have extensive post-transcriptional modifications of mRNA, primarily because transcription and translation are coupled processes. Bacterial cells lack the physical barrier of a nucleus, which allows transcription and translation machineries to function at the same time, enabling the concurrent translation of an mRNA while it is being transcribed within the system. The NUSG protein plays a critical role. NUSG has 3 separate domains and the functions of 2 of them are known. The NUSG N-terminal domain. NUSG NTD has the capacity to bind to RNAP, whereas the C-terminal domain NSUG CTD can combine with the NUSE RPase component of ribosomes. These 2 functions of NUSG enable transcription to be coupled with translation. NUSG CTD can also bind to RHO to terminate the transcription. This is the eukaryotic RNA processing. Hope you're taking screenshots. Go ahead with that. And In multicellular organisms, almost every cell contains the same genome, yet complex spatial and temporal diversity is observed in gene transcripts. This is achieved through multiple levels of processing leading from gene to protein, of which RNA processing is an essential stage. Following RNA of a gene by RNA polymerases to produce a primary mRNA transcript. Further processing is required to produce a stable and functional mature RNA product. This involves various processing steps, including RNA cleavage, specific sites, intron removing called splicing, which substantially increase the transcript repertoire, the addition of a 5' cap. Another crucial feature of the RNA processing of most genes is the generation of 3' ends through an initial endonucleolytic cleavage, followed in most cases by the addition of a poly-A tail, a process termed 3' and cleavage and polyadenylation. This is the illustration of 3' polyadenylation, and you have all the indicators here of the CPA complex subunits, right? And the details are polyadenylation is a required step for the correct termination of nearly all mRNA transcripts. In addition to determining the correct transcription length at transcription termination, the poly-A tail helps to ensure the translocation of the nascent RNA molecule from the nucleus to the cytoplasm, enhances translation efficiency, and acts as a signal feature for RNA degradation. RNA cleavage and polyadenylation is carried out by a multi-subunit 3' end processing complex, which involves over 80 trans-acting proteins comprised of the 4 core protein complexes, and polyadenylation signal sequences and the nascent mRNA initiate the cleavage and polyadenylation process, where approximately 50 to 100 adenosine residues are added to the mRNA. The location of polyadenylation. Right now, more than 70% of all genes have more than one polyadenylation signal, PAS. This gives rise to transcript isoforms differing at the mRNA 3' end, while alternative polyadenylation APA and 3 UTR changes in properties of the mRNA stability, localization, translation, internal pass usage, and introns of the coding sequence CDs changes the C termini of the encoded protein, resulting in different functional or regulatory properties. 5' cap formation. Now, in eukaryotes, the 5' cap found on the 5' end of mRNA molecule consists of a guanine nucleotide connected to mRNA via an unusual 5' to 5' triphosphate linkage. This guanosine is methylated on the 7th position directly from capping in vivo by a methyl transferase. It is referred to as a 7-methyl guanylate cap, abbreviated M7G. There are some other unusual 5' caps, but the 7-methyl guanate cap is the most common. For capping with 7-methyl guanylate, the capping enzyme complex CEC binds to RNA polymerase II before transcription starts. As soon as the 5' end of the new transcript emerges from RNA polymerase II, the CEC carries out the capping process. The enzymes for capping can only bind to RNA polymerase II that is engaging in mRNA transcription, ensuring specificity of the M7G cap almost entirely to mRNA. The 5' cap has 4 main functions: regulation of nuclear export, prevention of degradation by exonucleases, promotion of translation C ribosome, and translation promotion of 5' proximal intron excision. This is the diagram as mentioned there. In addition to the poly-A tail, nuclear transport of RNA is regulated by the cap by binding complex CBC, which binds to 7-methylguanylate capped RNA. And that's the diagram, which has an explanation below here. The CBC is then recognized by the nuclear pore complex and the mRNA exported. Once in the cytoplasm, after the pioneer round of translation, the CBC is replaced by the translation factors EIFOE and EIFOG of the EIF4F complex. This complex is then recognized by other translational initiation machinery, including the ribosome, aiding in translation efficiency. In addition, the 5' cap prevents degradation by functionally looking like the 3' end of the RNA and evading the degradation exon nucleases. Secondly, when the 5' cap is bound by translation machinery, it hides the cap from the decapping enzyme and increases the lifespan of the message. Decapping enzymes and degradation. All right, the decapping of a 7-methylguanylate capped mRNA is catalyzed by the decapping complex made up of at least DCP1 and DCP2, which must compete with EIFOE to bind the cap. The decapping enzymes are aided by enhanced proteins such as EDC3 that can bind with the mRNA in a sequence specific manner and help recruit the decapping enzymes. Thus, the 7-methylguanylate cap is a marker of an actively translating mRNA and is used by cells to regulate mRNA half-lives in response to new stimuli. During the decay process, mRNAs may be sent to P-bodies or granular foci within the cytoplasm that contain high levels of exonuclease activity. Now, the intron splicing. Eukaryotic organisms also have large tracts of non-coding regions interspersed throughout gene sequences known as intron sequences. These introns must be removed from the pre-messenger RNA before it translocates into the cytoplasm to undergo the translation process. Intron removal is mediated by the spliceosome, which is a macromolecular complex formed by 5 small ribonuclear proteins, SNRNPs, termed U1, U2, U4, U5, and U6 in approximately 200 proteins. Only the SNRNP molecules are shown in the diagram above. We have the intron splicing diagram here, right? Please take screenshots and make notes. If we take a closer look at the pre-mRNP that needs to be spliced, we can see that there are some key sequence elements within the intron sequence that enables these snRNA molecules to recognize the intron and choose the correct excision sites for the splicing. Exon 1 is shown here in blue and exon 2 in gray. There is a key GU sequence on this F5' side of the splice site and a key AG sequence on the 3' side that identify the borders of the intron. There is also a polypyrimidine sequence, PY, that is a key recognition sequence for the snRNP binding. There is also a branch point sequence that contains the critical adenine base that will be involved in the splicing reaction. Two trans-esterification reactions. The splicing reaction itself is a series of 2 trans-esterification reactions. So 2 trans-esterification reactions are shown here. So again, for the chemistry that will be occurring during the reaction, the key elements are the identification of the chiral cleavage sites at the 5', 3' ends of the intron, and the key adenine base that is found within the branch point sequence. The first trans-esterification occurs by positioning of the 2' OH residue of the branch point adenine near the 5' phosphate of the guanidine residue at the 5' edge of the intron. The 2' OH will mediate nucleophilic attack on the 5' phosphate group of the guanidine residue in the intron. And yes, this forms an oxyanion intermediate, and when the electrons rebound into reform the PO double bond, the 3' OH of the last guanidine residue in exon 1 would serve as the leaving group. Right, and we have the diagram here, which states that this forms the first intermediate product And the branch point adenine residue is now covalently linked to the 5' guanidine residue of the intron at the 2H position of the adenine. Let me get the diagram back for you. Have you seen that carefully? Okay, and just a second. Right, the final products are shown here. Exon 1 and exon 2 are joined and the intron has been successfully spliced out. The branch point adenine residue remains covalently linked to the 5' phosphate intron. This resulting loop structure is known as the lariat structure. A lariat is a rope used by cowboys to lasso or tether animals. The intron loop resembles this type of rope structure and why it has this name. See that? Lariat structure to loop or tether the animals. Intron splicing. We need to go back to the original diagram, see how the small nuclear RNP molecules are involved in the process. So intron splicing, complex A, U2AF, from here, complex or commitment complex. In the first step, you can see that there are a number of small proteins involved in the recognition of 3' intron body sequence, and that the U1 small nuclear RNP molecule recognizes and binds with the 5' edge of the intron sequences. This will happen simultaneously for all the 5' edges of all the introns within the pre-mRNP molecule. This is known as a commitment complex. Next, the U2 SNRP RNP will bind to the branch point and sequence. This is known as complex A. Intron splicing. Complex A is converted to complex B with the binding of 3 additional siRNA molecules and the loss of the SF1 protein. U4, U5 and the U6 join the complex and causes the folding of the mRNP so that the exon 1 will be in close proximity of exon 2. So there is no U3 SNRNP involved in this process. So researchers likely identified the SNRNP molecules before they knew their complete functions. So intron splicing. U1 and U4 dissociate from the complex. U2, U5 and U6 rearrange forming complex B. Intron splicing. Complex B quickly shifts to conform complex C, which is catalytically active and mediates the two transesterification reactions. Intron splicing, where once the transesterification reactions are complete, the U2, U5, U6 complexes releases the lariat product and the mRNA with the exons completely joined together, U2, U5 and U6, would then be recycled for another round of splicing. Alternative splicing. Alternative splicing offers an additional mechanism for regulating protein production and function. Options are determined by the expression of or exposure to introns elements present within unique cellular locations and environments. Additional sequence elements within the mRNA known as exonic and intronic splicing silencers are enhancers, ESS, ISS, ESC, and ISC respectively, participate in the regulation of AS. Specific RNA binding proteins including heterogeneous nuclear, ribonuclear proteins, HNRNPs, and serine arginine-rich SR proteins recognize these sequences to positively and negatively regulate AS. These regulators, together with an ever-increasing number of additional auxiliary factors, provide the basis for the specificity of this pre-mRNA processing event in different cellular locations within the body. Alternative splicing. You have exon skipping, alternative 3 SS selection, and alternative 5 SS selection, and the intron retention. There are several different types of AS events, all right, and which can be classified into 4 main subgroups. The first type is exon skipping, which is the major AS event in higher eukaryotes. In this type of event, a cassette exon is removed from the pre-mRNA. The second and third types, alternative 3 and 5 SS selections. These types of AS events occur when the splicer recognizes 2 or more splice sites at the end of an exon. The fourth type is intron retention, in which an intron remains in the mature mRNA transcript. This AS event is much more common in plants, fungi, and protozoa than in vertebrates, which is that intron splicing. Alternative, intron retention, in which intron remains a mature mRNA transcript, is more common in plants, fungi, and protozoa than in vertebrates. Alternative splicing, you have mutually exclusive exons, alternative promoters, alternative poly A. Other events that affect the transcript isoform outcome include mutually exclusive exons, alternative promoter usage, and alternative polyadenylation. Overall, there are many types of RNA modifications that help to regulate the lifespan and translation efficiency of a messenger RNA and help to control protein levels within a cell. In the next chapter, we'll look into more depth at the translation process. To give a recap on the RNA synthesis and processing, I'll just give you some notes now. The RNA synthesis and processing objectives: to understand the transcription process, to understand how RNA polymerase generates a new single-stranded RNA, to define promoters and promoter sequence, to describe RNA processing in eukaryotic cells. Transcription. The process of transcription synthesis of RNA synthesis directed by a DNA template is termed transcription and occurs in three phases: initiation, elongation, and termination. Please note every word by word, as these are very good short notes and pretty effective for the MCQs, so please don't miss out on any point that I'm reading out for you. Right, So in transcription, DNA is copied to RNA by an enzyme called RNA polymerase. Transcription to yield an mRNA is the first step of protein biosynthesis. Initiation of transcription. Promoter sequences. Unlike the initiation of replication, transcriptional initiation does not require a primer. Promoter sequences are responsible for directing RNA polymerase to initiate transcription at a particular point. Promoter sequences differ between prokaryotes and eukaryotes. In genetics, a promoter is a DNA sequence that enables a gene to be transcribed. The promoter is recognized by RNA polymerase, RNAP, which then initiates transcription. Prokaryotic promoters. The promoters for most prokaryotic genes have three sequence elements: initiation site, that's the start point, and transcription for most genes always starts at the same base, position 1. The start point is usually purine. Pribnow box lies 9 to 18 base pairs upstream of the start point. It's either identical to or very similar to the sequence TAT, that's TATAT. The Pribnow box is called 10 sequence box because it's usually found 10 base pairs upstream of the start point. The 35 sequence is a component of a typical prokaryote promoter. It is a TTGA called the 35 sequence because it is usually found 35 base pairs upstream of the start point. And the 35 sequence is a component of a typical prokaryote promoter. It is TTGA-CSA called 35 sequence because it is usually found 35 base pairs upstream of the start point. We then have the eukaryotic promoters. Each type of eukaryotic RNA polymerase uses a different promoter. The promoters used by RNA polymerase 1 and 2 are similar to the prokaryotic promoter in that they are upstream of the start point. However, the promoters used by RNA polymerase 3 are unique because they are usually downstream of the start point. And going to the next page. Initiation factors. Prokaryotic or karyotic sigma factor is required for accurate initiation of transcription. Eukaryotic initiation factors. The initiation of transcription in eukaryotes is considerably more complex than in prokaryotes. partly because of the increased complexity of eukaryotic RNA polymerase, and partly because of the diversity of their promoters. Elongation. The base required and fundamental mechanism of the elongation phase of RNA synthesis is the same in prokaryotes and eukaryotes. Template. A single strand of DNA acts as a template to direct the formation of complementary RNA during transcription. Substrates. The 4 nucleosides triphosphates are needed as substrates for RNA synthesis. Direction of synthesis. RNA chain growth proceeds in the 5-3 direction. Enzyme. Prokaryotes have a single RNA polymerase responsible for all cellular synthesis. The structure of RNA polymerase is complex. Large subunits, small subunits, sigma factors, etc. Eukaryotes have a 1 mitochondrial 3 nuclear RNA polymerase. The latter are distinct enzymes that function to synthesize different RNAs. Termination. In prokaryotes, there are 2 basic classes of termination events in prokaryotes. Intrinsic termination, row independent termination, involves terminator sequences within the RNA as it is being made that signal the RNA polymerase to stop. The terminator sequence is usually a palindromic DNA sequence that forms a hairpin. Row-dependent termination uses a termination factor called P factor to stop RNA synthesis at specific sites. When P factor reaches the RNAP, it causes RNAP to dissociate from the DNA, terminating transcription. Right? And the next would be a few illustrations here of the RNA polymerase holoenzyme binds non-specifically. The holoenzyme conducts a one-dimensional search for promoter, and when a promoter is found, the holoenzyme and the promoter form a closed complex. A conformational change from the closed complex to an open complex produces a transcription bubble. At the initiation site, a short stretch of RNA is then synthesized. The sigma subunit dissociation from the coenzyme and RNA polymerase clears the promoter. Accessory proteins, including NSA, bind to the polymerase. In eukaryotics, very little is known about how the termination transcription occurs. Now, this portion is basically just a sum up on what was being discussed. It's pretty effective if you read it through. Right. Next is post-transcriptional RNA processing. Once a gene transcript has been synthesized, numerous post-transcription modification or processing events may be needed before the transcript is functional. Prokaryotes. Post-transcription processing of RNA is not as extensive in prokaryotes as in eukaryotes. However, some processing does occur. Eukaryotes. Overall post-transcription processing is more extensive in eukaryotes than prokaryotes. This partly is due to the presence of a nucleus from which most RNAs must be transported. RNAs are processed during this transport. So just about winding up on this session of the lecture, and the remaining portions will be enumerated in the next lecture coming up shortly, right? So I just finish a few lines and that should be it. So the processing gives them the characteristics they need to be functional in the cytoplasm, such as increased stability of mRNAs, as well as allowing for another level of gene regulation. So the primary transcript, hnRNA, is capped at its 5' end, and a poly A tail 20 to 200 nucleotide in length is being added to the 3' end of the transcript, and splicing reactions remove introns and connect the exons, right? So that should be it for now. Please read through all details, take adequate screenshots and make your notes, right? Best wishes. I'll see you in the next lecture.